Hello everyone, let's see an interesting experiment to purify the plasmid DNA by alkaline lysis method. Take 1.5 milliliters of overnight incubated bacterial culture into a sterile microfuge tube. Then centrifuge at 10,000 revolutions per minute for 5 minutes. After centrifugation, remove the supernatant from the centrifuged bacterial culture and add 150 microliter of resuspension buffer solution into the tube containing pellets. After addition of resuspension buffer, vortex it for proper suspension of bacterial pellets. Now it's time for lysis. Add 200 microliter of lysis buffer into the tube containing resuspended pellets and close the tube tightly. Mix the content by inverting the tube for 4 to 5 times. And make it viscous. Now immediately add 150 microliter of neutralization buffer and mix it thoroughly by inverting 4 to 5 times and keep it on ice for 4 minutes. Again centrifuge for 10 minutes. Take out the supernatant and transfer it to another sterile microfuge tube. This supernatant may contain plasmid DNA. Then add equal volume of phenol chloroform into the microfuge tube containing supernatant. Mix the organic and aqueous phase by vortexing and centrifuge at maximum speed for 2 minutes at 4 degrees Celsius. After centrifugation, 2 different layers are observed. Transfer the upper layer into the new sterile microfuge tube. Add 2 volumes of ethanol into this tube at room temperature. Vortex the sample and stand for 2 minutes. Now collect the precipitated nucleic acid by centrifugation at maximum speed for 5 minutes at 4 degrees Celsius. Remove the supernatant completely. Now add 1 milliliter of 70% ethanol to the tube containing pellet and invert the closed tube several times. Recover the DNA by centrifugation at maximum speed for 2 minutes at 4 degrees Celsius. Remove the supernatant by gentle aspiration. Keep tube open at room temperature until ethanol evaporates. Dissolve the nucleic acid into nuclease-free water and vortex the solution of nucleic acid. Then store the nucleic acid solution at -20 degrees Celsius. Now perform agarose gel electrophoresis to confirm the purity of plasmid DNA. Observe the results obtained from electrophoresis. The image show the results of electrophoresis, and from this results we can conclude that plasmid DNA purified successfully.
In this experiment, we performed restriction digestion of PUC19 plasmid using EcoRI restriction enzyme. The assay buffer and BSA vials were thawed before use. The following reagents were added to a vial in a sequential manner. First, we added 41.5 microliter of nuclease-free water. Then we added 5 microliter of 10x assay buffer. To this, we added 0.5 microliter of 100x BSA. Now it was time for addition of the plasmid. 2 microliter of PUC19 was added to the vial containing the previously mentioned components, followed by which 1 microliter of the restriction enzyme EcoRI was added. In this way, the total volume was made up to 50 microliter. The vial was then incubated at 37 degrees Celsius for 2 hours in hot water bath. A sample was prepared for electrophoresis by addition of 20 microliter of the digested DNA in a different vial, and 4 microliter of loading dye, that is bromophenol blue, was added to it. The solution was mixed by pipetting and load on the gel. The gel was allowed to run for nearly an hour. The sample was run against an uncut DNA to confirm digestion. The uncut plasmid was added in the first well. Sample was loaded in well 4 and results were observed under UV transilluminator. A clear band was observed in well 4 indicating successful loading. The migration pattern and position suggested the plasmid to be nicked, verifying its digestion. In conclusion, the plasmid was successfully digested using EcoRI.
Hello everyone, welcome to today's experiment. In this session, we will perform a double restriction digestion of the PUC19 plasmid using BamHI and Ssp1 restriction enzymes. The PUC19 plasmid is a circular DNA molecule commonly used for DNA cloning. It features a multiple cloning region housing our first restriction site, specifically for the BamHI restriction enzyme. Notably, there are singular sites for both BamHI and Ssp1 in the plasmid, resulting in the generation of 2 segments upon digestion. These segments measure 604 base pairs and 2082 base pairs respectively. Now, let's walk through the experimental procedure. First, we added 11.8 ml of nuclease-free water to a vial. All components were kept on ice to maintain optimal reaction conditions. Next, 2 ml of 10x assay buffer, which was thawed before use, was added to maintain the pH of the reaction. Following that, 0.2 ml of 100x BSA was introduced. Now it's time to add the plasmid. We included 4 mol of PUC19 followed by the first restriction enzyme BamHI, that is 1 mol, and sequentially SSP1, that is 1 mol, to the vial. The vial was then incubated at 37 degrees Celsius in a hot water bath for 1 hour and 15 minutes. To prepare for electrophoresis, we combined 20 mol of digested DNA with 4 mol of loading dye. Our sample was loaded into well number 5 and run against a 1000 base pair DNA marker, pure uncut plasmid and double digested plasmid for confirmation. Bands corresponding to 604 base pairs and 2082 base pairs were observed, indicating a successful double digestion. Additional bands suggested potential incomplete digestion or variations in plasmid conformations. In conclusion, group number 4 successfully double digested the PUC19 plasmid using BamHI and Ssp1 restriction enzymes. Thank you for joining us in this
Experiment 4. Transforming bacterial cells using GFP cloning. We start by measurement of agar powder. We added 1.5 gram of agar powder in 100 ml of distilled water. Then we measured 12.5 gram of LB powder and added to 500 ml of distilled water. Next, we added 400 ml of LB to 6 grams of agar. The remaining 100 ml was added to 1.5 gram of agar in another conical flask. All the media was put in the autoclave chamber to make it sterile. This sterile media was then poured into petri plates and allowed to solidify. A ligation reaction was set up by addition of 11 microliters of water, 2 microliters of the vector DNA, that is 5 micrograms, 4 microliters of the insert DNA, that is 10 micrograms, and 2 microliters of the ligation buffer, which had 1x concentration. To this, we added 1 microliters of the T4 DNA ligase. This is the most important step of the reaction. The vial was pulsed to ensure settlement of all the components. It was then kept in the refrigerator at 4 degrees Celsius overnight. We took 50 ml sterile LB broth in a conical flask and inoculated it with colonies. This flask was kept at 37 degrees Celsius on a shaker for overnight incubation. The next day, the culture was divided into 2 tubes. These tubes were centrifuged at 6000 rpm for 10 minutes at 4 degrees Celsius. The supernatant was discarded while the pellets were retained. 1 ml of ice-cold calcium chloride was first added to the pellet, and after resuspension, we added 8 ml of ice-cold calcium chloride again. These tubes were then incubated on ice for 20 minutes. This was followed by centrifugation at 3500 rpm at 4 degrees Celsius for 20 minutes. The supernatant was discarded and 850 microliters of calcium chloride was again added to the vial. 100 ml of this suspension was allocated into 8 tubes. The following part of the experiment was performed by group 4. 100 microliters of competent cells were taken and added to a 1.5 ml sterile vial. Out of this, 5 microliters of competent cells were added to the vials containing the ligated and controlled DNA. The contents of the vials had been mixed by tapping and then placing on ice for 20 minutes. This can be marked as the beginning of the transformation process. Normally, the cells would be unable to take up the DNA as it cannot pass through the cell membrane. Hence, this step is followed by a heat shock treatment which creates transient pores in the cell membrane, allowing the DNA to enter the cell. This step is done by placing the cells in a hot water bath at 42 degrees Celsius for 2 minutes and then immediately placing on ice for 5 minutes. This is a very crucial step for ensuring a successful transformation. Following the heat shock treatment, 0.5 ml of LB broth was added to the vials and they were incubated at 37 degrees Celsius at shaker conditions for an hour. 100 ml of sample from the ligated and control vials had been spread on LB AMP plates labeled as test and control respectively. The plates were incubated overnight and observed under UV light at 312 nanometers. The following results were obtained and the colonies on the left plate can be seen to be fluorescent. These fluorescent colonies were counted and calculations for transformation efficiency were made. Transformation efficiency was calculated to be 2.86 into 10 raised to 5 CFU per μg. This concluded our experiment. Thank you.
Hello, we are going to discuss the practical for extraction and purification of DNA band from agarose gel. Let us look at the procedure. The DNA band was carefully excised from the gel. Excess gel was removed using sharp blades. The excised band was transferred to a fresh vial. The vial containing the gel piece was weighed and weight of the gel piece was calculated by subtracting the weight of an empty vial. The vial weighed 1 gram and the gel piece weighed 0.2 grams. 500 ml sodium iodide solution was added to this vial. This addition facilitates disruption of hydrogen bonds that hold the agarose polymer intact. This mixture was incubated at 35 degrees Celsius for around 2 minutes using a water bath. The agarose gel is dissolved and the DNA is separated from it. we added 15 ml silica solution. This solution contained silica beads to which the negatively charged DNA would bind. It was kept for 10 minutes. Then the vial was centrifuged at 12,000 rpm for 30 seconds. The beads formed a hard pellet and settled at the bottom. The supernatant containing the agarose was discarded carefully using a pipette and the pellet was retained. Then we added 200 ml washing buffer to this pellet. The washing buffer removes the unwanted negatively charged particles from the silica beads. The mixture was centrifuged at 12,000 rpm for 30 seconds. The supernatant was carefully discarded using a pipette and the pellet was retained. We again added 200 ml washing buffer to the pellet. Second addition was to ensure pure separation of DNA without any contaminants. The mixture was vortexed, due to which the loosely bound residues would easily separate from the beads. The vial was centrifuged at 12,000 rpm for 30 seconds. The supernatant was decanted carefully from the opposite side of the pellet. The vial containing pellet was incubated at 55 degrees Celsius for 10 minutes in a dry bath. After this, 25 ml 1xT buffer was added to the pellet. The 1xT buffer unbinds DNA from the silica beads and elutes it. The pellet is resuspended in the buffer by mild vortexing and centrifuged at 12,000 rpm for 1 minute. The supernatant is retained this time since it contains the DNA. The supernatant is centrifuged at 12,000 rpm for 1 minute. This helps to make sure that we got rid of all the beads and collected only the DNA. The supernatant is collected in another fresh and labeled vial. This is the purified DNA. Quantification was done using UV spectrophotometry. Thank you.
Experiment number 6, blue-white screening, performed by group 4. The cells used in this experiment were made competent by heat shock treatment. The following steps were performed. A loop full of overnight incubated culture was added to the LB broth. This suspension was then incubated at shaker conditions for 2 to 3 hours. The cells were then placed on ice for about half an hour. Following this, the culture was divided into 2 tubes equally and then centrifuged. The supernatant was then carefully discarded. Following this, 5 ml of ice-cold 0.1 molar calcium chloride was added in each tube. The tubes were then centrifuged and the supernatant was discarded again. Following this, a solution of calcium chloride and glycerol was added to the tubes. The glycerol is a cryopreservant and prevents the formation of nitrogen crystals in the tubes. Later, 100 ml of culture was collected in 3 1.5 ml vials. Following this, 100 ml of competent cells were added to 5 ml of the 2 different ligated samples in separate tubes. These samples were labeled as T1 and T2. Following this, a control tube was also prepared by addition of 100 ml of competent cells and 5 ml of the control sample. This sample did not contain the plasmid DNA. The tubes were then incubated on ice for 30 minutes. The incubation was followed by placing the tubes at 42 degrees Celsius for 90 seconds. After this, the tubes were immediately kept on ice for 10 minutes. After this, LB broth was added to the suspension. The tubes were then placed in shaker conditions for about 20 to 30 minutes. Succeeding this, blue-white screening could be performed. 100 ml of transformed and controlled cells were spread plated on X-gal IPTG lurea agar plates containing ampicillin. The following results were obtained. The control plate showed no growth, whereas the ligated sample 1 showed white colonies and ligated sample 2 showed blue colonies. White colonies confirmed the incorporation of the gene of interest. This concluded our experiment. Thank you.